Hello, my name is Professor Tom Wilkinson. It's a real pleasure for me to present the results of the Sprinter study, a randomized, double-blind, placebo-controlled phase 3 trial, which was designed to determine the efficacy and safety of inhaled interferon beta 1A for the treatment of patients hospitalized due to COVID-19. I was chief investigator on this study. I'm employed by the University of Southampton, but I've received research funding and consultancy fees from Synergen, with the biotech company that sponsored this study. There is a strong scientific rationale for the development of inhaled interferon beta as a treatment for viral-induced respiratory tract infections. Interferon beta is a naturally occurring protein which orchestrates the human cellular response to viruses. It is associated with a robust antiviral defense, and when deficient, is associated with worse outcome, whether that be in patients with chronic lung disease such as asthma or COPD, or associated with viral infections which can subvert this natural immune defense. Interferon beta has demonstrated robust antiviral activity in vitro systems, both to respiratory viruses such as flu, as shown in the middle panel here, or to SARS-CoV-2 infection early in the pandemic, where it was shown to be able to robustly suppress viral replication in vitro models. We feel that inhaled interferon is the appropriate route of delivery, as it optimizes delivery of the drug to the respiratory epithelium, which is the key target for respiratory viral replication. The phase 3 Sprinter study was based on the robust data of a phase 2 developmental study delivered early in the pandemic. Here, inhaled interferon beta was trialed in a hospitalized patient population and showed robust signals of efficacy and a good safety signal too. Here on the left, we can see the key results from the phase 2 study. This demonstrated very strong signals of reduction in progression to severe disease, improved time to recovery, and improved time to hospital discharge, with many of these findings in a relatively small phase 2 study showing significance. On the right here, we can see the data on recovery in graphical form, where we can see that the two arms of the study separate very early in the duration of treatment, with clear benefit of treatment in expediting recovery of the patients receiving the drug. This led to the design and delivery of Sprinter, the phase 3 developmental trial. The primary endpoints here were chosen as time to hospital discharge and a co-primary of time to recovery to no limitation of activities by day 28. The key secondary endpoints were also chosen, including the progression to severe disease, intubation or death. The power to deliver an assessment of the primary endpoints required at least 610 patients to be recruited into the study, and the trial was conducted therefore in 17 countries around the world, ultimately recruiting 623 patients who were randomized to drug or placebo between January and November in 2021. Here are the broad summary of the inclusion and exclusion criteria for the study. We included patients who were 18 years or older, were admitted to hospital due to the severity of their COVID-19, and who had tested positive for SARS-CoV-2 virus. They also had to require oxygen therapy delivered via nasal prongs or mask as an indication of the severity of their illness. The exclusion criteria are shown here on the right of the slide. Here are the patient demographics for the trial population. As you can see, this was a middle-aged population with a significant degree of comorbidity, which was representative of the population being admitted to hospital with COVID-19 at the time of the study. We also can see that a number of patients had received vaccination or indeed were treated with concurrent medications, including corticosteroids and antivirals during the trial. This is the step change from the phase 2 study where early in the pandemic, there was no vaccine exposure or concurrent medication used. Here's data from one of the primary endpoints, time to hospital discharge. And here we can see that there is indeed no difference between treatment and placebo for this primary endpoint, and the study did not meet either of the co-primary endpoints. However, if we look at some of the key secondary endpoints, we can see there are trends towards difference between the arms, although these do not reach statistical significance. Here we can see in progression to severe disease in both the intention to treat and per protocol populations, as well as progression to intubation or death within 35 days, there are trends in favour of the treatment arm in each of these outcomes. In order to explore the nature of this potential response, we looked to enrich the population of potential responders. And here we found that patients with more severe respiratory disease, as defined by a low oxygen saturation, less than 92%, or high respiratory rate, despite the use of supplemental oxygen at randomization, there seemed to be an enriched effect. This was particularly true in the per protocol population, which was defined largely by the criteria that patients had to receive at least 2 doses of the medication within the first 3 days of the study in order to adhere to the protocol criteria. And here we can see very interestingly that there seems to be an enriched treatment effect in this patient population that had more severe disease and received an adequate initial dose of treatment at least, with a statistical difference in the per protocol population seen here. In terms of safety, like in the phase 2 study, the phase 3 study demonstrated that the inhaled interferon beta was very well tolerated in the setting of COVID infection. The proportion of patients with any treatment emergent adverse events was very similar in treatment arm and placebo, and that the proportion of patients with any serious treatment emergent adverse events was actually lower numerically in the treatment arm. Interestingly, there was a numerically higher rate of pulmonary emboli in the placebo arm, with 5 in that arm versus 0 in the active treatment arm, which is an interesting observation that requires further research. So in summary, the study did not meet its primary endpoint looking at hospital discharge or recovery. This is potentially due to changes in standard of care, which the time patients were spending in hospital in the later phases of the pandemic had changed significantly from our earlier phase 2 study, with the use of vaccines and concurrent medications contributing to that difference. However, subgroup analysis increased our confidence that there may be a beneficial effect of the drug in certain populations, and we feel that there is therefore an opportunity to understand this effect further. Overall, this drug was very well tolerated, and there was a strong a positive safety signal both in this study and in the previous phase 2 trial. So overall, these clinical results provide a strong rationale to continue to investigate SNG-001 in further trials evaluating the effect in COVID-19, but also more widely in other respiratory infections where benefits may also be seen. I'd like to thank you for your attention to this presentation, and a big thank you to all members of the trial team who helped in its successful design and delivery. Thank you very much.